All right. Greetings, Earth and Space Explorers. Welcome to Doc Waller's Earth and Space Report for Friday, June 27, 2025. I'm your host, Bill Waller, an astronomer, science educator, and communicator, coming to you on a pleasant summer evening here in my hometown of Rockport, Massachusetts. These Earth and Space Reports are intended to engage and inform people like you who are curious about Earth as a planet, who care about our life-sustaining biosphere, and who wonder about the greater cosmos and all that it has engendered throughout space and time. Video recordings of these reports are archived on the Earth and Space Reports YouTube channel. They are also in regular rotation on the 1623 Studios Community Access Cable Television channel. I'm grateful for the continuing interest and support shown by Rockport educators and students, Cape Ann cultural colleagues, cohorts of the Gloucester Area Astronomy Club, myriad Earth and space scientists, educators, and communicators, far-flung friends, family, and other curious folk. Thank you for your active participation and encouragements. This evening, I'm delighted to be hosting Rockport's own Molly Twombly, more formally known as Kristen Twombly. Molly is a second-year PhD candidate at Tufts University in Medford, Massachusetts. Her research focus involves investigating the fragmentation patterns and pathways of biomarkers, more specifically adenosine, adenine, and ATP, as induced by UV radiation and oxychlorination in simulated Martian environments. Whew. In other words, she uses laboratory simulations to characterize the remnant biosignatures that we might find on Mars someday. I first got to know Molly as a student in my high school AP Physics class. She excelled in that class and went on to lead the science club that I hosted. Before graduating from RHS, Molly interned with me over the summer, where she helped me to implement an educator workshop in space exploration at Tufts University. She has since graduated from Norwich University, magna cum laude, where she majored in biochemistry. Nowadays, she's back at Tufts working at the crossroads of biochemistry and astrobiology. And that is what we will be hearing about this evening. So let's get going. The Zoom platform is all yours, Molly.
Thank you. Can everyone see my screen?
Yes.
Okay, perfect. Okay, so today or tonight, I'm going to be talking a little bit about looking for life on Mars, more specifically, the quantification of the degradation and fragmentation of adenosine under Martian conditions. So I know that's a lot to take in and it's kind of chemistry focused. So I've kind of made my talk more for a general audience. So if you have more targeted questions, if you'll just wait till the end, that would be awesome. So I know Dr. Waller kind of went on this, but who am I? I'm a second year PhD candidate at Tufts University. I'm in the Kunavis Planetary Chemical Analysis and Astrobiology Lab. I'm also a 2019 graduate from RHS. And I work on two projects currently right now, biomarkers. I work with adenine derivatives. And then I'm also recently on a MICA project, which is microfluidic icy world chemistry analyzer. But for all this talk, I'm going to be talking about the biomarkers for the adenine derivatives. So just a little quick overview. I'm going to be looking for life on Mars, adenosine as a biomarker, some short-term and long-term goals, and then get a little bit into the experimentation that we do in our lab. So since ancient times, humans have looked up at the sky and wondered our place in the cosmos. And one of the main questions that everyone asks at some point in their life is, are we alone in the universe? And so this was first discussed in the 19th century. And this is from a McKay paper in 2010. And we see it a lot through pop culture as well. H.G. Wells' The War of the Worlds in 1898. A little more hopeful for humanity is The Martian in 2015. And it's also referenced across songs as well, like David Bowie and Elton John songs. And so Mars is the most promising location in our solar system for finding past or present life. And this is because of its proximity to Earth. There's evidence of past water, and there's also similar early conditions to Earth. And so discovering life beyond Earth would profoundly impact science, philosophy, and exploration. But of course, we always come back to that fundamental question is, does extraterrestrial life resemble life on Earth? Where should we look and how should we look? So this is one of my favorite photos. This is from NASA Goddard Space Flight Center, or the Big Think, and this is an image of early Mars and Mars today. And at first glance, you're probably like, that looks like Earth. That looks like a picture of Earth today and Mars today, but that's actually early Mars. So both planets, Earth and Mars, early Earth and early Mars, had abundant liquid water, a carbon dioxide rich atmosphere, and intense volcanic activity, creating the perfect setting for this prebiotic chemistry. These shared conditions suggest that if life could arise on Earth, it might have also emerged on Mars through this process called convergent chemical evolution. This is where similar environments drive the development of similar biochemical processes. So, if Earth had the essential building blocks for life, water, carbon, nitrogen, and phosphates, then Mars also had the essential building blocks for life, water, carbon, nitrogen, and phosphates. And it could have also had energy-rich environments like hydrothermal systems and redox gradients that could power these life-sustaining reactions. Minerals on both planets as well, like clays and iron oxides, could have catalyzed the formation of complex organic molecules. However, as we know, Mars lost its magnetic field, causing its atmosphere to thin and its surface water to disappear. This is perhaps what halted these life's progress. I also want to add somewhat tangentially that by studying Mars, we're not just searching for aliens, we're also investigating whether the chemistry of life is a universal consequence of the right conditions, or whether Earth's story is a cosmic norm or an extraordinary exception. So when looking for life, we look at these molecules. We look for biosignatures. And so when there are no signs for life as we know it, we can look for chemical signs. And so we investigate biomarkers. And so a biomarker, which is short for biological marker, is any measurable substance, structure, or process that indicates the presence of life, past or present. In this context of astrobiology and the search for extraterrestrial life, biomarkers are crucial clues that scientists look for to determine if life exists or existed on planets, moons, and celestial bodies. So the biomarker that I'm most interested in is adenosine. I also work with adenine and ATP, adenosine triphosphate. And so these are nucleosides for RNA, which are essential for biological functions. Adenosine has roles in energy transfer, blood flow, and brain function. It's also a component of ATP, which is life's energy currency in the body. And so biotic adenosine is produced in living systems. And as I just mentioned, it's critical for life processes, but abiotic adenosine can also be produced, hypothetically be produced, or experimentally produced outside biological contents, which is significant for understanding life's origins or potential extraterrestrial life. So in abiotic environments, such as in prebiotic chemistry, where life's building blocks may have been formed through these natural processes like UV radiation or catalytic interactions on mineral surfaces, adenosine is essential to life on Earth because it plays critical roles in genetics and energy transfer. As a nucleoside, adenosine is aromatic and it consists of the adenine base, which is right here. I don't know if you can see my mouse, but it's up there. And then it has the ribose sugar, which is down here. Is a fundamental building block of DNA and RNA, as I had mentioned. Additionally, adenosine forms ATP, which is the primary energy carrier. ATP fuels nearly every cellular reaction, every cellular function that happens throughout your body, from metabolism to protein synthesis. All living things, animal, plants, and microbes need adenosine because it is integral to cell division, muscle contraction, and nerve signaling in animals, photosynthesis and growth in plants, and it also plays an essential role in metabolic functions in microbes. So, moreover, adenosine's role in the genetic code also makes it indispensable. Adenosine is really important in replication and transcription, processes that are critical for cell growth and reproduction. So, therefore, when we're trying to think of these undeniable signs of life, we think of DNA because all living things have DNA. So, adenosine is therefore a perfect contender for ubiquitous signs of life on Mars. And so, of course, when looking for life on Mars, we've sent missions to Mars. So, just a quick overview of all of these missions. We, as humans, have sent around 55 missions to Mars as of 2024. There's been some successes and there's also been some failures, but key rovers such as Curiosity and Perseverance have transformed our understanding. There's also been some organic compounds found, such as thiophenes and benzenes and long chain alkanes, which were found in sediments really recently. That was the Fresnet paper that was just published. And then we've also seen modern liquid possibilities. There's subglacial lakes and briny liquids that may have harbored microbial life or potentially could harbor microbial life today. So the first mission that was sent was the Viking landers in 1976. And so Viking conducted groundbreaking biology tests yielding ubiquitous results later attributed to the perchlorate oxidizing organics during soil heating. And I'll talk about that in a little bit. But Viking really shaped future missions because it forced NASA to develop non-destructive tools such as Curiosity's SAM and deep drilling strategies to bypass these surface oxidants. And then in 2007, NASA launched Phoenix, and it was the first detection of perchlorate, which is a 0.6 weight percent, via the WetChem lab or the WICL. It revealed the high ClO4 Cl minus ratios, hinting at active surface chemistry. And its legacy really explained Viking's false negative organic results due to the salt content on the surface. And of course, we can't talk about missions to Mars without mentioning Curiosity, which was in 2012 and is still present. And so Curiosity has a sample analysis at Mars, which is the SAM, which is the onboard suite, which is actually 3 instruments working together. So it has a quadruple mass spectrometer, which analyzes the composition of Martian gases. It has a gas chromatograph or a GC, which separates and identifies different organic molecules. And then it also has a tunable laser spectrometer or a TLS, which measures isotopic ratios, which can provide insights into the origin and history of the Martian materials. So with all of those rovers and landers on the surface of Mars, you would think that we would have found huge organic molecules like intact, perfect adenosine, perfect signs of life, but that's not the case. So this is a schematic proposed by Carrier and Cunaves in 2015, and this shows a perchlorate schematic. So at the electron level, perchlorates are destroying biomarkers through a series of oxidative electron transfer reactions. So perchlorate itself, the ClOx, is highly oxidized form of chlorine, where chlorine is in the +7 oxidation state, meaning it has a strong tendency to accept electrons and then be reduced. So when heated or exposed to radiation, perchlorates decompose into reactive intermediates like chlorate or oxygen radicals and chlorine radicals through this homolytic cleavage. So it's actually breaking the molecule apart. These radicals are electron deficient and highly reactive due to their unpaired electrons. So when they encounter organic molecules, they're abstracting electrons, especially from electron-rich areas like the CH or CC or CN bonds that adenosine is full of. And so this initiates a chain reaction of bond breaking and then thus fragmenting the complex organic molecules into their smaller oxidized products. So if that weren't bad enough, perchlorate actually has a really high reduction potential, which makes these electron transfer reactions thermodynamically favorable. So it's driving the destruction of these biomarkers. So this electron-driven breakdown could potentially explain why detecting intact organic molecules on Mars is so challenging, because any exposed biomolecules are rapidly oxidized and they're degraded by these highly reactive species. And so just to walk through a little bit of these reactions, we have a Cl minus, which will go to a ClO3 minus, and then this can go all the way to the perchlorate, which is the ClO4. So this O3, the carbon trioxide, is oxidizing the chloride, which is the Cl minus, to chlorate, and then all the way to perchlorate. And then if we look on the mineral surface here, we have the Cl minerals, which could be just regular salt, NaCl, which will go all the way to ClO4 minerals. And then we have organics and the oxidants, which could be any variation of the ClO4 minus, the ClO2, the Cl radical, the O4 radical, which will go all the way to the carbon dioxide and water. So if this is happening and this destruction is destroying any organics, how are we able to find life? How are we able to find signs of past life. So in a sense, how do we simulate these processes? So this is just a little brief walkthrough of what we're looking for. So we're looking at a sample preparation. We have a Mars simulation chamber that we use. We then want to do a solid phase extraction. We then need to do an analysis through mass spectrometry. We then want to identify any fragments, and then we can characterize the whole biomarker at the end by using the fragments to kind of build it together like a puzzle. So I'll talk a little bit about this sample preparation and the Mars simulation chamber. So this is an overview of how we prep our samples. So we actually have a physical Mars simulation chamber. So since we're a chemistry lab, this is all done physically, not through a computer simulation. I know when you think, oh, a Mars simulation, you're thinking it can be done on a computer, but we actually have a chamber physically where all of this is taking place. So a sample is plated with a SOL mixture, which is of silicon dioxide, SiO2, ethanol, and water, and it's plated on the bottom of a beaker and then left out to evaporate. And this simulates the soil on Mars or the surface on Mars. Then we're using a sodium perchlorate and ethanol solution. So that's then left to evaporate, simulating the salt on the surface. And then adenosine in solution is then plated on top of the SOL layer and the perchlorate layer to simulate the organics that would have been deposited on top of the salt on top of the soil. And so in our Mars simulation chamber, we have a UV lamp on top, and that simulates the sun. And so these are the parameters that we use. We use a pressure of 7 mbar, a UV exposure of 300 watts, and then we have a gas mixture, which is trace amounts of oxygen, argon, and nitrogen, and then a CO2 balance. And then I do all of my exposure times, 24, 48, and 72 hours. Those are just what I'm starting with at the moment. And then prior work in our lab has shown that the samples that we're running fall in the range of 200 to 280 nanometers for wavelength for the light, which is within the ultraviolet, the UV portion of the electromagnetic spectrum, which is specifically in the UVC range. And so this is part of the light that comes from the sun. So we're really trying to simulate what it would be like to be an organic on the surface of Mars. And so this is an overview of some of the samples and controls that I run. So we run plain adenosine, we run adenosine plus UV, we have adenosine, UV, and the salt, and then we have adenosine, UV, the salt, and the SOL. And so eventually the goal is to go all the way up to the real meteorite sample, which is EETA 79001, which is a meteorite sample that we have from Mars. And so the goal is to simulate it in Earth conditions, Martian conditions, with or without UV, with or without the salt, and then with or without the soil simulant, so that we can have a baseline for what the fragmentation patterns look like under all of those conditions. And so then once we have our sample and it's been run in the chamber for these time variants of 24, 48, or 72 hours, we need to extract the biomolecule and its fragments out of the perchlorate and SOL solutions. So we have a solid phase extraction or an SPE method. So we have a column, which is a Hypersep C18 column, and we're using a lure lock system in order to filter out that SiO2 SOL mixture because it's really damaging to the instruments that we use. And so we're conditioning the column with a methanol and water, which will just ensure that the column is uniformly wet, which will activate the sorbent, which is the filter at the bottom, and remove any residual manufacturing impurities. We use the water because it will balance the hydrophobic properties of the stationary phase, which is the sorbent, and prepares the column for loading the aqueous sample. So then into our sample, we have the adenosine perchlorate and SOL all mixed together post-chamber. And so that's going to go through, and we're going to use that lure lock again to filter out the SOL. Then in our column, we have the adenosine and perchlorate left, and we're going to try to elute any contaminants with this. So we're going to use NH4OH to get rid of the perchlorate. And then we're really looking for the adenosine in any of its fragments. So then we're going to use a 2% acetic acid solution to elute that adenosine for an analysis. And so, like I just said, we will have the analysis next, which is the mass spectrometry analysis and the fragment identification. So there's a few things we can do post-chamber analysis. So the first thing I'm going to talk about is an LCMS. And we use an LCMS TOF system, which merges this liquid chromatography separation powered with the time of flight mass spectrometry's precision. So liquid chromatography mass spectrometry, making it an ideal tool for hunting molecular clues for life. So imagine this as a molecular detective that first uses liquid chromatography, which is the LC, to separate the complex mixtures, like the Martian samples, into individual components, much like sorting a box of mixed puzzle pieces by their shapes and by their colors. So when searching for these biomarkers, the system ionizes these separated compounds, which is typically via electrospray ionization, which gently gives each molecule an electrical charge. So think of like if you're spraying like Febreze or perfume, it creates a spray. And so then that sends them racing down the time of flight, the TOF, the analyzer's flight tube, which is right here. So here's the key for life detection. The different biomarker fragments, like the ribose or the sugar, are like molecular runners in a race. So the lighter fragments will sprint ahead while the heavier ones are going to lag behind with their exact mass to charge ratio, or their M over Z, determined by their finished time. And we call that the elution time. So this system is incredible. It has a less than 5 ppm accuracy, meaning it can detect mass differences as small as 0.0005%. And so this becomes extremely crucial when distinguishing between potential biomarkers and similar looking molecules that might arise from non-biological processes, or contaminants that we might see in the instrument, or something in the blank, or something that is left over from our sample prep. So this precision allows us to identify true biosignatures with confidence, even in extremely old samples, or potentially in the Martian samples where the biomarkers may exist at trace levels among the parent molecule that we're seeing intact. And so also in liquid chromatography on the right here, we use a gradient. It's a carefully timed ramp that helps separate different chemicals in the sample. So also, instead of using the same solvent strength throughout the run, the system gradually mixes a weak solvent such as water with a stronger one, acetonitrile, or this ACN. And so early on, weakly bonded compounds tend to separate out, while the stronger solvents later in the gradient help push out the more stubborn molecules that want to hang on to our column. And so this makes the analysis sharper, faster, and more efficient. And so here are some of my results from the LCMS. So this is a control EIC, which is an extracted ion chromatograph, where I'm looking at just the M over Z for adenosine. So this is the 268.16 M over Z, compared to a 24-hour EIC, which is a 24-hour irradiated Martian sample extracted at that same 268 M over Z to look for the adenosine. So if you look closely, you can see that from going between 24, 48, and 72 hours, we're seeing a strong decrease in concentration of the adenosine sample. So just for example, on this one right here, we have the control, and then down here, we have a 24-hour sample, and the abundance has changed, meaning that the parent adenosine has degraded over time. And so then we can also compare the total ion chromatographs to the 24-hour samples. So this is again a total ion chromatograph of the control adenosine. So this is just the whole run without the specific adenosine sampled out, control compared to a 24, 48, and 72 hour. So here you can see that the total ions are also decreasing after 24 hours, 48 hours, and 72 hours, not just the adenosine itself, but all of the other molecules in the sample. And so we can look even closer at this. So we can look at a control TIC versus a 24-hour TIC, and we can look at where the peaks are happening. So if you look at the green, that's our control, and then you compare it to the orange, we're seeing a new little peak right down here, and then we're seeing a disappearance of this big peak right here. And so that could potentially be a new fragment peak that's forming. And we can do the same thing for a 24-hour compared to a 48-hour. So the 24-hour is up here in orange and the 48-hour is down here in purple. And we can clearly see that this peak right here is gone. This peak right here is gone. And then this peak is dipping even further where we're not seeing a dip there. So not only after 24 hours are we seeing a difference, we're seeing another decrease and almost disappearance of these fragment peaks from 24 hours to 48 hours. It doesn't stop there. We can do it again. We can do it for the 24-hour TIC to the 72-hour TIC. So again, the 24 is in orange and the 72 is in yellow. And we're seeing a disappearance of this fragment peak. We're seeing a disappearance of this fragment peak. And then we're seeing a little bit of an appearance of a fragment peak here, which could potentially be a fragment peak. But that's all great. We can see that the concentration is decreasing and we can see that there are peaks, but how actually are we going to identify these peaks? So unfortunately, our LCMS in our department does not have a database in it because we just don't have the model of that instrument. So we need to come up with another way to actually plug in our data to an instrument and have it tell us what it's seeing. Kind of like what we were talking about at the beginning with AI, but we can have an instrument do it. So this is a GCMS, and this is a gas chromatography mass spectrometry, which is a sophisticated analytical technique that combines two powerful methods to separate, identify, and quantify chemical components in these complex matrices. So first, gas chromatography, or the GC, separates the components of a sample and by vaporizing them and carrying them through a long, thin column using an inert gas. So as the molecules travel through this column at different speeds, they're traveling based off of their chemical properties and interactions with the column's coating. They emerge one by one, effectively getting sorted for individual analysis. And so these separated components then enter the mass spectrometer, which is the MS, just like we had on the LC, where they are ionized, typically by electron impact or chemical ionization, so EI or CI. And they're broken into characteristic fragments, and then they're detected based on their mass to charge ratio, just like that M over Z that we were seeing at the LCMS. The resulting mass spectra serve as unique fingerprints that help identify each molecule with high precision. But that sounds too easy. So there always has to be a caveat. So, however, many biologically relevant compounds, such as adenosine, are polar or thermally unstable, making them difficult to analyze directly by a GCMS. So they may decompose under these high temperatures or they stick to the column, leading to poor separation and weak detection. So this is where a process called derivatization becomes essential. So by treating these compounds, either pre-chamber or post-chamber, with reagents like BSTFA, we can chemically modify them, replacing reactive hydrogen atoms, such as the OH or the NH2 or the COOH, with trimethylsilyl, which is the TMS groups. So this transformation makes the molecules more volatile, thermally stable, and easier to detect, and we can actually run them through the GCMS now. So this derivatization not only improves chromatographic separation, but also enhances the quality of the mass spectra, providing clearer signals and more reliable identification. So this is a derivatized adenosine at 4-TMS. So we can see that the TMS, the silyl groups right here, have replaced that 4 active hydrogen sites. So we have one here, 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 and here. And so when this takes place, we can actually run our adenosine post-chamber or pre-chamber through the GCMS, and we get really good separation results. So we're seeing this peak right here at about 12.8 minutes. That is this adenosine for TMS. And so this is just a blank adenosine, a stock adenosine, not irradiated in the chamber. This is just straight up from the bottle adenosine mixed in ethanol and then run through. But we can also run adenosine with derivatization in the GCMS post chamber. And so this is what a Mars chamber sample would look like. So we have adenosine coming out at about 8 minutes here. This is with derivatization. And then the abundance over here is quite high. So we know that we're getting quite good separation, and all of these peaks are separated. And so the whole point of this is to start identifying fragments. So we have this little fragment peak right here at like 6.3 minutes, and then we have a fragment peak here at about 4.2 minutes. And so by doing consecutive runs, we run over and over again in triplicate, we have these possible fragments. And so at our 4.2, we have this fragment, which is a dimethyl primido. It's a TMS derivative of some kind. And then we also have a 3-TMS derivative on this side. And if we remember what the adenosine looked like, if we go back to this slide, we see that there's the sugar and then we see the adenine base. And we can kind of see that we have somewhat of an adenine base right here. And there's a five-membered ring in there that could be part of the sugar. And then we come over here and we're seeing that 6-membered ring that could be a part of it. We're seeing nitrogen groups. So these look like possible fragments that we're seeing from post-Mars chamber fragmented adenosine in the Mars chamber. And so we do this and we run over and over and over again to confirm the validity of our results. And so what does all of this mean, putting it all together? So essentially, we want to create a catalog of fragments. We want to be able to say, this is what happens to adenosine. This is how adenosine fragments when you run it at 24 hours under these conditions where you have sol, UV, and perchlorate, where you have sol, UV, and not perchlorate, where you have sol and perchlorate, but no UV. And then we want to run all the way up until we get to this replicate with the meteorite sample. And so essentially, when we have future missions to Mars, or if there's ever a sample return, we can look for soil work. So we have a list of what the organics are fragmenting into, so we can compare them against what the rovers and landers are seeing in the actual soil. And we can look at the building blocks and we can say, oh, that pieces together to this, that could be adenosine. And so some future work that we have is we always are doing post-chamber analysis of this adenosine. We're seeing this ion peak comparison. We always have to run blanks, controls, and then also irradiation time. We're doing peak area comparisons to see how much of our parent adenosine is fragmenting and degrading. How much are we actually losing? There's always GCMS, which has the database, the NIST database that we're comparing our fragments to, and always updating that because we want to make sure that we're not seeing any column bleed or contamination. I also work with adenine and ATP, so I'm always in the process of doing GCMS, HPLC, which is high performance liquid chromatography, and UV-Vis method development for both of these compounds. And then, like I was mentioning, there's this theory called molecular assembly theory, which is to come in later, and this is IR and NMR. But essentially, molecular assembly theory is you're taking these fragments and you're literally building them back together like Legos to form your parent ion to see what's going to happen when your parent ion fragments, what fragments can you piece together that you're finding in the simulation to get back to your parent molecule. So that's a very brief overview of and very quick overview of what I do with the biomarker project. But another project that we're working on is this MICA, and this is the microfluidic icy world chemistry analyzer, which is a miniaturized laboratory instrument designed to access the habitability potential of ocean worlds like Europa and Enceladus by analyzing the chemical composition of their liquid water environments. So this really builds upon the success that NASA had with the Wet Chem Lab from NASA, the Phoenix mission. And so MICA employs microfluidic technology to process extremely small sample volumes while measuring critical habitability indicators such as dissolved ions. So this could be Na+, K+, Cl-, SO4 2-, etc. pH levels, oxidation reduction potentials, and conductivity through an array of specialized electrochemical sensors. And so Enceladus was selected for this because it represents a significant technological advancement in astrobiology by enabling these detailed chemical characterizations of icy world environments that could potentially harbor life while also supporting comparative planetary science studies of ocean chemistry across different celestial bodies. And so that's also really important to me, being from Rockport, growing up on the ocean. Doing a comparison of the ocean chemistry is very cool to me. So these are just some people that I would like to thank. Of course, Sam, my advisor, I'd like to thank him. Bernadette, thank you for joining. And then I have two undergrads that worked for me this past year, Lion Kim and Emma Smith. She just graduated and she got a job out in California. And then, of course, our funding sources, NASA and Tufts. And then just have some references and thank you. And I'll take any questions you have.
All right. Let me get the gang.
I
Uh,
can see that there are some messages in the
The
chat
gang's
too.
all here. I got a question, if you don't mind. Do you have
Sure.
to take gravity into consideration, the difference between Earth's gravity and Mars's gravity in any of your experiments or chemical reactions?
Right now we're not. We're just looking at the surface. We're looking more chemically at how the biomarker is fragmenting. Maybe gravity will have to come in when we actually put boots on the ground, if we ever put boots on the ground. But right now we're not taking that into account.
Okay, thanks.
um, I have a question um, about UV radiation. You seem to always have it as a destructive agent uh, on Mars and simulating it. Um, is there a case for the UV radiation actually uh, catalyzing uh, other chemical constructs?
Yeah, adenosine has this cool thing where I'm seeing that it actually dimerizes, which is where it's forming these protective groups. So when the UV radiation is hitting that parent molecule, it actually wants to protect itself. So it could be a situation where the UV is helping and forming just a different kind of biomolecule, but it's still the adenosine being affected by the UV.
Okay.
And other groups have also shown that with other compounds. So it's not just adenosine that has this dimerizing effect where UV is actually protecting these molecules.
Okay.
How much stronger is the sun's effect on Mars than it is on Earth through the atmospheres?
It's quite stronger. I think that I mentioned it a little bit in my talk. We haven't done the flux of our lamp, but we know that there's variations in it so far. Um,
I
but
thought
I
300
I
watts
think
was a lot of power.
what'd you
In
say?
your experiment, the 300 watts of UV, that's pretty intense.
Yeah, we haven't measured that it's falling between the 200 and the 280, which is what other grad students had done in our lab, which is the UVC range. So that is matching what we're seeing um on Earth right now.
Thanks.
Molly, what is the deepest that the rovers have penetrated the soil to get samples? How far down?
I think they've only gone a few inches to my knowledge. There was Phoenix that did that in the when they like reach their arm down and they just like scoop.
Mhm.
I don't think they've dug any deeper than just
Yeah.
what the arm can move.
Isn't it probable that the deeper under the surface that uh, organic life could possibly exist, that it might be better preserved?
Yes. So we have another grad student. That's actually what Bernadette does in our lab. She works on permeation, where we're
Mhm.
looking at how far the chlorine dioxide gas can penetrate the surface and what it's going to do to organics the deeper that you go down. So it could be that when you go down deeper, there's not as much fragmentation and the biomolecules are preserved, and we will see signs of past life if we dig.
Good. Thank you.
Mhm. Yes.
Hi, Michael.
Do we still have unretrieved samples on Mars? And is
Yeah.
it a question of funding?
Yeah.
Okay, that's what I thought.
Yeah.
And is there anything in particular that would these samples are holding up as far as your research? Are there any things you are looking for with the return of these samples ultimately that would help?
Yeah, I know that there's a proposal out there in the works somewhere for sample return. I know that it will be really hard to get the samples back. it would be awesome if we could get them back. But I think that the main thing would just be looking at the soil composition to try to look for any of the fragments that we're seeing. But I'm not sure when they're planning on bringing anything back.
I think Michael Deneen had a question.
Yeah, I was muted still. Sorry. Molly, your PhD work is tied pretty specifically to a group of funding choices. Do you keep that in mind as you decide how specific you want to make your skill set?
Well, I want to work for NASA, so I definitely chose my grad school and my lab based on that. So my funding is very specific because I hope to work there one day,
Right.
but we'll see how it goes right now with the current situation and everything with how funding is being received and all that.
Are you looking over your
chaos.
shoulder? Does that affect your choices at all?
No, this is my dream.
Good.
So
Looking ahead.
yeah,
Yeah.
looking for brighter days ahead, definitely.
I have kind of I don't know, a technical question. I noticed that in your processing, you used ethanol and methanol as part of your various treatments. And I'm just wondering whether those organic molecules play any chemical role, or are they chemically inconsequential?
So we're just using methanol to I use methanol to do my SPE method, so it actually all gets filtered out, so it's not interacting with the adenosine because we're pushing it through the column, so it's only activating that sorbent. And then because adenosine is soluble in both, if it were to get stuck, it would just come out with our wash and our elutions is what it's called from the column. But I also run like when it goes to waste, I run the waste on the instrument to make sure that I'm not collecting any adenosine in either of those samples. So right now it doesn't appear to be holding on to either of those. But the ethanol is just used in the sole preparation, and that evaporates off, so you're just left with dry SiO2 on the beaker.
I see. Hmm. Um, I wonder whether there's any of these alcohols on the surface of Mars.
Yeah, I'm sure there's something. There's some kind of alkyl chain or something, or carboxylic acid.
Hmm. And one last question. Hydrogen cyanide seems to be the go-to molecule for a lot of astrochemistry leading to astrobiology. And I'm just wondering whether that has been something you have been looking for or simulating. Is there any of it in existence on Mars at this point in time?
That's not something that I've looked into for my research. I've just been solely focused on the adenine derivatives, but that's definitely something that we could look into, but I haven't encountered any of that.
Okay. Seems like nitrogen is the major player in a lot of
Yeah,
this.
I definitely.
I
So,
have a, I have a
Let me
question that isn't directly related to your research, but it certainly set up the environment that you're studying. What caused Mars to lose its magnetosphere? Do
Its
we?
atmosphere.
Yeah, its atmosphere and its internal magnetic field or magnetic dynamo.
I think it was a combination of collision, and then it also has something to do with the core. I'm not a
The
geologist
core
or a
solidified, right?
geophysicist, but
yeah,
Mm.
Yeah.
yeah. It's
yeah.
low mass, so things
no,
cool down very quickly.
Yeah, much more so than Earth, where we
no.
still have a liquid
Right.
core. Mm.
Yeah, that's
Let me
my
make
Let me just sneak
geology.
in one here. I know we got a minute. I have visions of panspermia running into Mars. How do you know if you get a piece of something really interesting, where it came from? Did it come from a Russian probe? Did it come from a meteor? Did it come from 2 inches under the surface on a Martian trilobite?
Yeah, that's what makes it so hard.
Sourcing.
Yeah. Right now, because nothing's come back, we don't have to worry about that. But I'm sure once they do a sample return, or if they do another mission where they're going specifically to retrieve and come back, they'll pick their site accordingly.
How important are the Martian meteorites that have been retrieved and identified?
We're going to use those down the line because we want to try to simulate the surface as closely as we can. And so we're going to use the ET0091 that I had mentioned.
Mhm.
So I'm just not there yet. This is only my second year. It takes a while to run all of the samples overnight and for 24, 48, and 72 hours, and then do all the analysis. And most of it's done by hand too at this stage. So
Wow. Well, I'm glad you're doing it and not me, because I'd take naps. Any other questions?
So
I
cameras.
have one. Molly,
Mhm.
knock knock.
Oh,
Who's
no.
there?
Mars polar lander.
Mars polar lander who?
Exactly. Well, this was great.
Tip your waitresses.
Yeah.
So no conclusion as to whether one could grow potatoes with poop in the Martian soil.
Yeah, no conclusions, but definitely an optimistic outlook.
You're not running that experiment?
No. Good movie though.
It's a great movie.
Good book too.
Hmm.
We had a woman that actually gave a lecture about that heating soil experiment on Mars, the earliest ones that they were trying to extract soil and then heat it up and look. So she gave a lecture maybe, I don't know, 3 years ago. You guys remember
A
any of
while
that?
ago. The Viking.
I bought her
Yeah.
book.
Yeah,
She
the
had a
Viking
lot
twin
of
lander.
horse. She learned how to ride a lot of horses back then. She was in the horse
Mm-hmm. Yeah, in Pasadena.
club. Yeah,
We have a
yeah.
cryochemistry program at DAC next month with a PhD student from CFA. You might be interested in.
Yeah, join us.
Yeah.
We're an
Okay.
eclectic group.
Well, if we don't have any other questions or comments, I'm going to give Molly a hearty thanks. I'm
Thank you for
so
having
delighted
me.
to see you
Great.
doing such great things in Sam Kunave's lab with Bernadette and the others. And may you continue to fight the good fight.
Yes.
Continue
Thanks
to
for
get
having me.
funded.
Have a good
you.
weekend, everybody.
Yeah.
Keep cool.
Okay, thank you.
Wasn't
All
Thanks
easy.
a
right.
lot, Artie.
Go
Good night,
well.
everybody. Thank you, Bill.
Nighty night.
Right.
Thank you, everyone.